Alrighty, well, hello everyone, Professor Kyle here, and today we are going to be covering histology, which is the content of Lab 2. So we're going to be discussing topics such as cells and tissues and everything that comes along with it. Again, the function of this video and others like it is just to provide you with a condensed version of the lecture component from our lab class. And this specifically is going to be basically a crash course version of it because I know if you are viewing this, you are probably prepping for a practicum exam. So you're trying to get all of the highlights, get everything nice and consolidated into your memory in your head so that you can write it down when it comes to the exam day. With that being said, we're going to go ahead and get started. And first things first, if we're talking about cells and tissues, well, what's the generalized structure of the cell? Well, we have that listed right here. First and foremost, please commit this illustration as well as the major highlights on the subsequent slide, which gives you the basic function of many of the structures to memory. Commit this to memory. There's a matching exercise that you can do with your online textbook. I would highly recommend doing that if you don't feel confident in identifying and spelling out the names of all of these structures. But let's talk about some of these structures right here before we get too ahead of ourselves. I want to focus in right here on the nucleus, right? And we all know the nucleus is the site of where your genes are, where your DNA and your chromosomes are. Within the nucleus, we have this structure called the nucleolus. Okay, so it's its own organelle within the nucleus, but it's not membrane bound like many of the others out here. The function of the nucleolus is pertinent to the production of these things called ribosomes, which is the first point here on our next slide, ribosomes. And the special thing I want you guys to remember about ribosomes is this is the fundamental site of protein synthesis. This is where the mRNA strands that are produced from the DNA inside of the nucleus are translated into protein chains, right? And so the ribosome is where that process occurs. Most typically, it occurs on the surface of this next organelle that we're going to discuss called the endoplasmic reticulum, or the ER. The ER is subdivided into two components, as we see listed right here. Okay, so we have the rough ER, which is the portion of the ER that is studded with those ribosomes, giving it, again, a rough, rippled structure. And that is, again, the site of protein synthesis, as well as once it is within the lumen of the ER, there is some additional post-transcriptional modification that occurs to the protein, some folding and that sort of stuff, addition of functional groups. The second half of the ER is called the smooth ER, the smooth endoplasmic reticulum. And so again, rough ER, I want you to think protein synthesis. Smooth ER is too many slides, is the site of liposynthesis or lipid synthesis and steroid production. Steroid is a somewhat scary word in pop culture. It essentially just means molecules that are based off of cholesterol. So again, main distinction I really want to emphasize, rough ER is studded with ribosomes on its surface, site of protein synthesis. Smooth ER is for essentially fat-related cells, lipid production, as well as cholesterol. Next, we're going to talk about the Golgi apparatus. We see that here in the lower right-hand portion, this green guy right here. The Golgi apparatus, for all intents and purposes, is essentially like the postal service of the cell. So it is responsible for packaging proteins, enzymes, other acids, that sort of deal, and then sending it off to wherever its respective destination is, whether that be another organelle, for example, the mitochondria, or even for outbound secretion, such as with these little vesicles right here, peroxisomes and lysosomes. Additional post-transcriptional modification of proteins also occurs within the Golgi, but main thing I want you to think of is that sort of functional aspect of packaging and sending off functional units. For example, such as lysosomes and peroxisomes. And we're not going to get into the weeds as to the constituent parts of these. They're defined based off of their function as well as the composition of the enzymes and acids that they contain. But essentially, lysosomes and peroxisomes, bundle them into your head as being essentially the recycling bins within inside of the cell. So these are the mechanisms by which the cell can break down organelles that are no longer functional, that it has no need of and it wishes to break down into its constituent parts. They also serve an immunological function for your immune cells. Think of like your white blood cells, because they can, as we can see right here, be secreted into like the extracellular space to break down the membranes of potentially foreign invaders, such as bacteria or viruses. Okay. Next, we have the mitochondria, and we all remember from high school biology that the mitochondria is the powerhouse of the cell. It's the powerhouse in the sense that it produces this incredibly important molecule called ATP, adenosine triphosphate, and that is the most fundamental energy molecule within inside of your cell. We are constantly breaking off that third phosphate group, the triphosphate, and converting ATP into ADP and utilizing the energy produced therein to accomplish cellular function. It's a pretty incredible stuff. Next, we have the centrioles, and centrioles are going to be very relevant to what we discuss next, which is going to be the cell cycle. And they are responsible for the formation of this stuff called the mitotic spindle. It's this framework of very similar to kind of the composition of extracellular cytoskeletal structures such as these microtubules and filaments. But it is responsible, these centrioles are responsible for organizing, arranging, and separating chromosomes during cellular replication, or mitosis, as we will come to see. And lastly, we have some cytoskeletal elements that we see listed here. Just note that that is what these are right here. We're not going to go any further than that. Again, this is anatomy and physio, not a biology, cellular biology class. So now we're going to launch off into this cell cycle, mitosis, it being the most fundamental form. We're not going to talk about meiosis, which is, think of like your eggs and your sperm, that sort of stuff. So mitosis is essentially the division of a single cell into two daughter cells. And we can break this up into various different phases. First and foremost, we have this space right here, the 3/4 of our circular graph here called interphase. Functionally, the vast majority of the cells in your body spend the vast majority of their time in interphase. This is where the cell is just doing its thing. It's whatever its physiological function is, it's doing it. Specifically in G1, kind of the growth phase of it. At some point, it receives a signal that says, hey, I need you to prepare to turn into 2 cells and not be 1 anymore. At that point, it enters S phase of the subphase of interphase, where it is continuing to grow in size, duplicating its cellular machinery, its organelles, mitochondria, that sort of stuff. But most importantly, it's undergoing the process of DNA synthesis. So it is duplicating its DNA. It's going from a state of N1, 1 copy of its genome, to N2, 2 copies of its DNA. Then we have G2. G2 is a relatively short proportional to the prior two, especially when compared to G1. This is essentially like a checkpoint phase. The cell is basically evaluating whether or not it successfully duplicated its DNA, and if there are any fatal issues, the cell may commit apoptosis. It may off itself to prevent the development of cancers. Assuming everything is going smoothly, it will then enter the mitotic phase, M phase, which has several subcomponents that we are going to discuss. So here we see interphase. Okay, the most important thing to note here is that we have our nucleus, and our nucleus is consolidated, but the internal DNA is not consolidated into chromosomes. It's kind of loosely organized as chromatin. We can also identify that nucleolus that I mentioned earlier, which is responsible for ribosome production. So again, prophase is just an illustration. So now what we are seeing, we are proceeding down this path right here. After interphase, once we're in process of mitosis, we enter prophase. The distinguishing facet here is we can now see consolidated chromosomes, okay, these chromatid pairs. DNA can exist in 2 states. It can exist in a stretched out state called euchromatin, where it is easily accessible for transcription to produce mRNA strands, which will then be translated to produce protein strands. And conversely, instead of being loose, it can be consolidated, which is called heterochromatin. Okay, so loose is euchromatin, consolidated into this chromosome structure is called heterochromatin. And we can see those centrioles are now starting to migrate to the distal portions of the cell in preparation for pulling apart, which then leads us quite logically to metaphase. So now in metaphase, we can see that the chromosomes are clearly lined up along the medial portion of the cell and are preparing for separation. Take a look at that over here. We can see prophase. Early prophase, we're starting to see some consolidation of the chromosomes, and then by late prophase, we can start to identify individual chromatid pairs right here. Okay, and then here's that metaphase. Again, the chromosomes are oriented along the middle. The centrioles would be located right here. We can see those mitotic spindles that we were mentioning earlier as well, emanating from them, and they are physically pushing and connecting to the chromosomes at the midline. So metaphase, middle. That's your strong hint right there. Next, we have anaphase, which is the process. You will now see that the chromosomes are separating into sister chromatids. The centromeres split apart the spindle fibers, pulling the chromatids to what is about to be separate daughter cells. Telophase is the process of cytokinesis. Cyto means cell, kinesis means movement. And so we can see the formation of two sister daughter cells. We're also seeing that furrowing occur as they are beginning to finally close off their membranes and separate into two individual cells. This is a review sheet from the textbook. We're not going to cover that right now. Again, this is crash course. Alrighty, this table is also in your lab handout from class. This is everything that you need to know when it comes to identifying histological slides. So knowing the tissue, where the tissue is from, what type of cell it represents, and what the tissue function is what you are responsible for. If you get this table down pat and are able to recognize all of the cells associated with it and tissues associated with it, then you are prepared for your practicum exam. We'll take each of these in turn in terms of the general classifications. Excuse me. Wrong button. It was a bleep button. Alrighty. So we have 4 basic cell tissue types, but before I break down this and we proceed down our various types of tissues, I want to just orient us in terms of the hierarchical structure of cells to tissues, organs, all that sort of stuff. So the most fundamental living unit, as we all know, is the cell, right? And cells that are organized for a common purpose are the basis of a tissue. Cells organized for a common purpose. If we have tissues that are organized for a common purpose, then that is an organ. Organs, likewise, that are organized for a common purpose are organ systems. And the culmination of all those organ systems is you, right? Does that make sense? Now we're ready to hop into this. So we have 4 main classifications of tissue types that we need to discuss. The first and foremost of which, not foremost, just the first of which is epithelial tissue. The most basic function is covering, or separating would also be a equivalent, perhaps better description, but covering and separating different areas, whether it be within inside the human body or separating the body from the outside world. So for example, your skin, right? Then we have connective tissue. Connective tissue is by far the most broad category. It has a wide variety of things. As we will see, everything from your bones to your blood is technically falling under the category of connective tissue. Then we have muscle tissue, which makes sense. That's how we're accomplishing movement. That's how we're accomplishing these levering motions. And then nervous tissue, which is obviously regulating and controlling the whole system. So let's start off with epithelial tissue. When it comes to naming and categorizing epithelial tissue, there are 2 primary characteristics that we are going to focus on. We have simple and stratified. The distinction there is a simple epithelia is a single cell layer thick. And stratified epithelium, conversely, has multiple layers. We can see that on the subsequent slide here. So this would be a simple tissue because it's just 1 cell layer thick with a basal surface touching probably to some form of basement membrane. And then we have a stratified tissue right here with multiple of the same cell type stacked on top of each other. Secondly, we can look at the structure, the shape of the cell to give us an indication as to what category it should belong in. Squamous is going to be flattened or scale-like. Think of like a pancake sort of a shape. This will also be reflected in the shape of the nucleus. The nucleus will also be flattened out. Next, we have cuboidal, which are relatively round or cube-shaped, as we will see. We'll see that most evidently in the inside of the kidney glomeruli later on a histological slide. We'll also note that the nuclei of these cells are relatively round as well. And then we have columnar, which are fairly rectangularly shaped and have slightly elongated nuclei. Again, this will all make more sense as we look at it. Now, obviously, when we look at tissues and we have stacked cells in squamous tissues, we might note that the shape of the cells varies throughout the course of that layering process. So how do we name them? Well, we look at the apical surface. We look at the outermost, the top layer, and the shape of that cell dictates the naming convention for that tissue. Again, here we see squamous, flattened nucleus, cuboidal, relatively symmetrical, symmetrically proportioned with a round nucleus, and then columnar, rectangularly shaped, slightly elongated nucleus. We also see that written here verbally on this slide. Here are some examples of these cells in different tissues. Here we have the innermost portion of your lung. This is the alveoli of the lung, which are single cell layers thick, simple squamous epithelium. This is very beneficial for the process of diffusion, right? We're trying to exchange gases across a membrane. Well, the thinner that membrane is, the faster that exchange can occur. Next, we have simple cuboidal. This is looking at the inner lumen of a kidney. We'll have some subsequent slides that zoom in a little bit further, but these little round tunnels you see right here are simple cuboidal cells, and they're roughly symmetrical in shape. And then lastly, right here, we have simple columnar epithelium inside of the colon, inside of your large intestine. And we can see these cells are all nicely uniform. The nuclei are roughly adjacent to each other. They're not stacked. Emphasis on the not stacked idea. We'll see that being very useful and relevant later. Cool. Breaking these down further, let's look at each of the cell types and a general physiological function. So again, we've already discussed simple squamous is one cell layer thick. This is a even more zoomed in form of the alveoli of the lung. And this is where gas exchange would be occurring to take in oxygen and expel out carbon dioxide, right? We also see it in certain parts of the kidney glomeruli, not the filtration portion, which we said was simple cuboidal, but kind of the lining of the glomeruli. Again, that function of separating things. We'll see simple squamous there. And there's some other examples here, but emphasis on diffusion being the function. Here we see the lumen of a blood vessel. We have red blood cells floating along inside the lumen of the vessel. And then again, that notion of separation or of covering things, we have connective tissue out here. So this simple squamous epithelium is separating the connective tissue from the lumen of this blood vessel, right? This tiny little layer right here. If I was to outline the cells, like this would be, oh, no, that's a bad line. This would be a cell. This is a cell. This is a cell. That's a cell. Makes sense? Sorry, my drawing is not very good. Next, we have simple cuboidal epithelium, right? So these are the filtration units inside of the kidney. This is a much more zoomed in version from what we saw on the prior slide. Here, we can definitely see the delineation of these roughly symmetrical cells here with relatively round nuclei. That should be a dead giveaway as to what you're looking at. And this is where we would go through the process of, again, filtering our blood for things we want to excrete, such as urea and other substances. Formation of urine being the excreted out byproduct of that process. Then we have simple columnar. Here we're looking inside of some portion of the intestine. Where does it say this is? It doesn't specify. But inner lining of the small intestine. So here we can see these are our cell structures, right? Makes sense. And notice that the nuclei are all relatively along the same line. We also see some smatterings of other cell types right here. We have a goblet cell producing mucus that is then secreting out into the lumen of the intestine. These cell types are commonly seen for the function of absorption. So if we were to zoom really, really far in on this, because this is already probably a 400x magnification, we would see that we have this little brush border, these tiny little cells lining the edge right here. I'm sorry, these tiny little structures lining the edges of the cells right here. The function of these microvilli, they essentially look like this on the apical surface of the cell, right? So we can imagine the rest of the cell bodies down here. We have all these little protrusions in the membrane along the edge. The function of that is to increase the surface area so you can absorb more nutrients across the membrane. If the membrane was just flat, then this would be our limiting factor for absorption of nutrients. But if we increase the surface area, now we have all these different angles from which we can absorb nutrients. Make sense? I know my illustrations are amazing. Alrighty. Anything else we need to note here? No, it should be good. Here's another slide. I think we actually took this in the lab, which is why it's not as good, but it's still very useful because this is very representative for what you might see on the practicum. So again, be able to recognize the cell type, where it is likely from, and what that associated function would be. So here we're thinking absorption. Next, we have pseudostratified. So previously, I kept emphasizing with columnar, with simple columnar, that the nuclei were relatively in the same plane, right? Here, we notice that that's not the case. This tissue is called pseudostratified because it looks like there's a stacking of multiple cells on top of each other. However, that is not the case, hence why it's called pseudo. If we were to examine all of these cells in their entirety, we would notice that each one of these cells at some point has attachment to this basement membrane right here. So this guy's attached, as well as even like this guy is attached, okay? Again, we have some goblet cells mixed in here. Where are we looking when we see pseudostratified? Well, right now we're looking at the lining of the trachea of your windpipe. We have a mixture of these goblet cells along with pseudostratified epithelia that are capped off on their apical surface by these guys called cilia. Now, what is the physiological function of this complex arrangement of cells? The goblet cells produce mucus, which they then secrete and then floats up to here to the top surface of the cilia. And then all of these cilia are going to wave and do this motion in a unified direction. In this case, it would be upwards towards the esophagus, where you would then swallow the mucus. You do this periodically, oftentimes without even noticing. The function of that mucus is to pick up any particulates that are on their way down into your lungs. We want to prevent dust and bacteria from getting to the alveoli because, as we saw before, they're single cell layer thick. They're very sensitive tissues and would be a very desirable site for infiltration for bacteria and viruses and such. So by lining the trachea with mucus and air flowing this way, we can catch some of those particulates, and then the mucus travels up through the trachea, is swallowed into your stomach, and then all of it gets dissolved away, and you get to breathe healthily, right? For individuals, you may have seen an individual who is smoking have like a hacking or a coughing fit, and you may wonder, why is that the case? Well, for chronic smokers, what tends to happen is due to the adverse effects of smoke inhalation, these cilia, I'm going to delete all of my lines here. It's getting a little bit chaotic. These cilia end up dying for individuals who are chronic smokers, meaning that they're no longer pushing that mucus upwards towards your esophagus so that you can swallow it. So the only way to clear their windpipe, to clear their trachea, is to to mechanically cough and get that mucus up to the surface to swallow. It's a fun little side fact. I'm not going to go on too many tangents, but that's just a pretty interesting one. Here we have some more examples of pseudostratified epithelia. This was taken from one of our slides in the lab. Certainly this one was. I don't know about this one. This one definitely was. I think actually I took this, me or one of the other faculty in the lab. And so we can see that sort of pseudostratification occurring because we're seeing nuclei that look like they're stacked on each other, but really all of the cells are adjacent in the end. Alrighty, next we have stratified squamous epithelium. There's two kind of subcategories of stratified squamous epithelium based on, first of all, their location, and second of all, the presence or absence of a protein called keratin in their structure. Okay. Instances like this where we do not see the proliferation of keratin, as well as I think on the next slide. Yeah, this is the inner lining of the vagina. We notice that these are very good at resisting kind of mechanical abrasion. They're also fairly stretchy because they can flatten out. And due to there not being keratin, they're fairly smooth and viscous, so things can slide along it, which would be very beneficial in your esophagus, right, where you're trying to swallow things. We can see non-keratinized in the esophagus, mouth, and vagina. However, conversely, we see keratinized in your skin, right? So the presence of keratin in the cell cytoplasm, which eventually ends up with the cell dying, think of it, it's very structurally similar to like the composition of your nail beds, or your nails or your hair. And it's basically just a mechanical reinforcement to the cell to resist any sort of rough motion, cutting, that sort of stuff. So again, multiple cell layers thick. The mitotically active ones, the stem cells, are down here in this lower layer. And then as we can see, there's a stratification. These cells, cell tissue types can only get so thick with living cells because our vasculature is down over here. These epithelia tissue are not vascularized. They don't have their own blood supply. They rely on diffusion from adjacent blood vessels to get their nutrients. So the thicker this layer gets, the farther away cells become from their source of nutrients. So they can only get so thick while remaining living. Here's just another example. I believe we took this in the lab. Stratified squamous of the vagina. So again, mitotically active cells with their little basement membrane right here, form of connective tissue that they're attaching to, and then cells grow out that way. Just another picture. All right, transitional epithelium. A little bit of a weird one. Where do we see this? Well, we only see it in one area. It looks similar to stratified squamous, but we see it on the inner lining of the urinal canal and the bladder. And it's very good for resisting stretch. So we see connective tissue down here. We have a basement membrane running along here. And then we have the stacking of cells that would be fairly effective at stretching out like that, right? How do we distinguish this? I know because you're all worried about practicum. How do we distinguish this from stratified squamous epithelia, which looks fairly similar? Well, one thing you'll note is that there's a relative rounding to the apical cells here that we don't typically see in stratified squamous. So if you see this sort of rounding, this sort of healthy cell shape at the apical surface, you're probably looking at transitional epithelium. So it stretches readily, and we see it lining the urethra, urinary canal. All right. Connective tissue. All right. Connective tissue is a fairly large category of tissue types. Everything from things like the tendons and ligaments between your muscles, cartilage, buffering like your meniscus, buffering your bones and joints, your bones themselves, as well as your blood are all technically in the same category. Now, you might think to yourself, that is just a random categorization by lazy scientists, but that is not the case. All these things do share certain features of function in common. Additionally, to kind of, encourage you and reassure you that there is a basis for this, it's a fun fact to note that all of these tissues originate from the same early embryological layer called the mesenchyme. So during early embryological development, there are 3 layers that form. All of these tissues and their associated cell types originate from the same mesenchymal layer. Alrighty, so subcategories of connective tissue. We have loose connective tissue and we have dense. Oh my goodness, that was a loud Windows update notification. We have loose connective and we have dense connective tissue. And then we have regular and irregular subtypes of dense connective. We'll take each of these in turn because when we see their physiological function and where they're located, it makes a lot more sense as to what they are. An important note, because we're going to see there's lots of similarities between like connective tissue and cartilage and that sort of stuff. And students can sometimes get a bit confused as to the differences between the two because there's overlap in their structure. But one key difference is that connective tissue proper, the originating cells producing this matrix of material are fibroblasts. When we're talking about cartilage later, we'll be talking about chondrocytes. So fibroblasts are connective tissue proper, and chondrocytes are for cartilage. Here we're talking about loose connective tissue or of the areolar subtype. Where do we commonly see this? Well, there is a layer of this immediately under the lining of your epidermis of your skin. There's multiple components to the particular layer under your epidermis, but the first layer immediately under it, forming the basement membrane, is the lamina propria, and that is areolar connective tissue. It contains a variety of different cells, macrophages to phagocyze bacteria, and also just support and resist the stretching motion that obviously your skin is frequently subjected to. In terms of the composition of the fibers, we see several of our most important fiber types. These pink strands are collagen fibers, and then these purpley dying ones that kind of look like little spindles, little pen marks almost, are elastic fibers. Each of these fibers serves different physiological functions. Collagen fibers are very resilient to both compressive and stretching forces, so they are very strong forms of reinforcement. Elastic fibers, on the other hand, are very good at resisting recoil, or in other words, they are able to stretch and then return back to their original shape quite well. So you can imagine the mixing of that would be very good for something that's going to be subjected to a lot of pinching, rolling, and impact forces such as your skin. Cool. Again, we want to be able to recognize the tissue, the components of the tissue, and a basic physiological function. Those are the things we really want to emphasize here. Next, we're talking about adipose tissue. In other words, fat cells and fat tissue. So yes, fat is technically a form of connective tissue. Provides reserve energy, right? Fuel storage, food storage in the form of these lipid droplets, these fat droplets right here. So an important note in terms of looking at the histological slides, the actual portion of what we would consider the normal cell is this thin lining surrounding, you can see here, surrounding a fat droplet, okay? So all of this loose space here, this is not a part of the cytoplasm as you would typically think of it, the space that is occupied by organelles and that sort of stuff. The actual cell is, again, I'm kind of taking a guess here. The actual cell body and cell cytoplasm would be this space right here, but then it has a portion of its membrane that's wrapping around each of these fat droplets. We see this in a variety of locations. We see it in the subcutaneous layer under your skin, as well as around reproductive organs. So, for example, like the mammillary glands on a breast, as well as protecting reproductive organs in female anatomy inside the abdominal cavity, and a variety of other locations. It also can provide some form of thermal insulation, right, to prevent heat loss, and again, supports and protects internal organs. Okay, so here we can see a histological slide of adipose tissue, and as we can see here, it does not dye very well. It does not stain very well, but we can note some particular structures here. So again, this would be the lipid droplet, and these are portions of the cell cytoplasms that we're seeing right here, okay? They're just not staining particularly well. But if you saw this on a slide, you'd be able to recognize it as adipose tissue, food storage, thermal insulation, mechanical protection, that sort of stuff would be physiological functions. Do not mistake it for simple squamous epithelium in the lining of the lung. That is a common mistake. So these very large open spaces are lipid droplets, and compare that back to the simple squamous epithelium over here. Did I already pass it? Yes. This is the inner lining of the alveoli of the lung. So this is for gas exchange. Okay, so just note the differences there. Alrighty, dense connective tissue, dense regular connective tissue. Where do we see this? Well, we see it with the tendons and ligaments connecting your bones to muscle and muscles to muscles. Note that we have a uniform structure or a regular orientation to these fibers. These are predominated by collagen fibers. There might be some reticulars in there, but the primary matrix of fibers produced by the fibroblasts in this tissue type are going to be collagen fibers, because really we're not focused on stretching here. You can imagine, again, a little bit of a physiological tangent. If we have a muscle and we want that muscle to contract to accomplish movement, right? We don't want the connection points between that and the muscle or the bones that we are levering against to be stretchy, because if that bond ends up stretching, then the muscle would need to contract even further to accomplish the same levering motion, right? Whereas if this connective tissue is very sturdy and resists that sort of stretching motion, then all of the contractility of the muscle fiber will translate into movement of the joint, right? So we don't want to see a lot of stretching occurring in this, hence why it's mostly collagen fibers. Again, tendons, most ligaments, that sort of stuff. Attachment muscles to bones or muscles to muscles and bones to bones. Okay. Here we can see from a tendon, again, we notice that there's a relative uniformity in the orientation of these cells and the collagen fibers within. Now we have elastic fiber. Okay, so complete opposite end of the spectrum from what we just discussed. With tendons and ligaments, we don't want them stretching very much because we're trying to accomplish movement, mechanical motion. But with elastic tissue, elastic fibers are very stretchy. They're very good at resisting recoil, right? So they like to stretch and then return back to their original shape. Where would this be physiologically beneficial? Well, we would see this as being very beneficial in the aorta. So here we have the heart. And imagine the heart, you're ventricles are pumping blood. Every time that heart contracts and pumps blood, it's going to dramatically increase the hydrostatic pressure to push blood, shunt blood into your vasculature, into your circulation. And every time that happens, excuse me as I erase things, every time that happens, the hydrostatic pressure in the aorta is going to increase substantially. And so we want that to be able to accommodate a certain degree of stretching. But then after that stretching, when the ventricle is done contracting, return back to its original shape. So that's a very important example of where we would see elastic tissue. We also see it mixed in with some other fiber types in certain areas. For example, ligaments associated with the vertebral column, as well as others, bronchiotubules that need to dilate but then return back to their original shape. Yeah, pulsating blood flow through arteries, etc. Then we have the passive recoil of the lungs. I'm going off on a bit of tangents here, but when you breathe in deep and then you exhale, when you breathe in deep, you're pushing against the elastic force of the lining of the lungs themselves. And when you exhale, that natural desire to return back to its original shape that you've stretched past is part of the process of breathing out. That makes breathing out easier. Okay, this is the lining of the aorta, okay? So relatively uniform fibers, but very stretchy. Now we're going to talk about cartilage, okay? Everything thus far, I want you to categorize into fibroblasts and into connective tissue proper. Now we're going to talk about cartilage, okay? So we're seeing some similar terms here. We see elastic cartilage. So this is elastic cartilage, not elastic fibers from connective tissue proper. And the cells responsible for the production of cartilage are chondrocytes. So connective tissue proper or fibroblasts. Cartilage is chondrocytes. We'll see the chondrocytes, I believe, on the next slide here. Yes, right here. Oh my gosh, my lines are just so terrible. I am so sorry. I play video games with a controller, not with mouse and keyboard, so you can see evidence of that here. We have three categories of cartilage, three main types. We have hyaline cartilage, by far the most abundant. We see it intermixed in all sorts of areas within the human body. Then we have elastic cartilage, similar, but with more elastic fibers. Okay, more elastic fibers, so it's going to be a bit more stretchy and able to return back to shape. And then we have fibrocartilage. Okay, what are some examples of where we're going to see these things? Well, let's start off with fibrocartilage, which is kind of an intermediary between hyaline and dense regular tissue. We're going to see this in vertebral columns, so the vertebral discs in your spinal column, and then in like the meniscus of the knee and other sorts of things. What does it look like? Well, it looks something like this. So here we can see chondrocytes. These are individual cells that are inside of little hollow spaces that they've made for themselves inside of the matrix that they're now essentially entombed in, called lacunae, which basically translates to lakes. Lakes are holes in Latin. And so again, we see this in the intervertebral discs. So we want to have fairly strong, we have a mixing of elastic as well as elastic properties, as well as some of the properties of hyaline cartilage. And that's because we want to resist mechanical motion, but also be able to stretch and flex a little bit. Anything else we need to know here? No, we can see there's a bit of a higher concentration of what would be elastic fibers in these darker staining areas. And then this blue stuff is going to be more of the collagen fibers, collagen matrix here. But obviously there's a mixing of both, which is why we state that fibrocartilage is a mix of hyaline and dense regular connective tissue. Hi, alrighty. So I paused the recording to add a couple more slides because I realized I didn't have examples of hyaline and elastic cartilage in the PowerPoint, which would obviously be very useful since this is a histology lecture and is potentially material on the test. So just to quickly reiterate, so we have the chondrocytes and the lacunae, and then here we have these blue staining fibers are primarily collagen. And it looks like we have a fairly high density of what looks like elastic fibers up in here. If it's more purple, it's probably a higher density of elastic fibers. The blue staining stuff is collagen. This is a histological slide, looks kind of cool. You can see all of our individual chondrocytes over here. Here's an example of hyaline cartilage. Again, it is interspersed widely throughout the human body. We see hyaline cartilage at the endpoints of our long bones to help reduce friction in our joints. We also see it here mediating a couple different tissue types. The one I'll note is, so this entire section here is hyaline cartilage. Then we can see individual little lacunae and chondrocytes is these little circles, cells in here. And then this portion down over here, this is adipose tissue, right? So this is just a branch of cartilage running adjacent to and through adipose tissue, again providing it some structural rigidity, because adipose tissue by itself wouldn't have much form or structure. Continuing on, here is a relatively zoomed in picture of elastic cartilage. So the main distinguishing facet is, again, all of these purple staining bits. This is high density of elastic fibers, elastic cartilage. And again, this is going to be good at resisting stretching sorts of motions. So to distinguish those, fibrocartilage, relatively blue staining, maybe some little bits of purple. That's probably fibrocartilage due to the presence and high concentration of collagen fibers. Hyaline cartilage is going to stain relatively more clear and more pinkish, probably adjacent to some other tissue types because it's fairly widely dispersed. And then elastic cartilage going to stain very purple because of all of the elastic fibers. All right, continuing on with bone, also known as osseous tissue. Supports and protects body structures, makes sense. Stores fat and synthesizes blood cells. The fat portion may not be super intuitive. This is the yellow bone marrow. That's typically at the end points of your long bones. And then we have more collagen than compared to cartilage. Sure. Has inorganic calcium salts. So going back to kind of the storage function, bone. So yes, there is a certain degree of fat storage, but bone is also very important for like mineral homeostasis. So the calcium phosphate that forms the primary mineral structure of ossified bone can be broken down periodically if you have extremely low blood calcium levels. Then your body will adapt to that by breaking down bits of bone to then bring its calcium levels back up. Osteocytes are the main cells responsible for bone production and maintenance inside of bone tissue. There's subtypes: osteoblasts, osteoclasts. We'll get a little bit into that, but not too heavily. We'll reserve that for your more in-depth bio classes. And then osteons are the individual structural units. What does that mean? So this would be an individual osteon right here, this kind of circular structure, right? And then within which we have a central canal, which can accommodate, is vascularized. It can accommodate blood cells. And then here we have individual lacunae. The lamella is just kind of the space in between the in between these osteons that isn't uniformly encompassed. But inside here, these are the lacunae where individual cells, osteoclasts or osteocytes, are residing. So we all know bones in general function. This is what they look like. We'll leave it at that for now. I could go on into physiological function and then, Wolff's law and bone reinforcement, that sort of stuff, but it's not relevant for our exam. This is a histological slide. So we can see again, here is an osteon right here. Okay. Cylindrical structure. These are individual cells in the matrix. Blood doesn't sound like a tissue. It is. Think of it as, so just as with, way back here, apologize for the slide jump around. So just as with connective tissue, we have this ground substance. This is a relatively gel-like matrix. So if you were to slice this in part, it would have a gel-like consistency, obviously then reinforced by collagen fibers and elastic fibers. It would have a fairly solidified structure. But this ground substance produced by these fibroblasts is a relatively viscous or gel-like sort of a substance, and that forms the extracellular matrix for connective tissue. Fast forwarding back to blood, well, the equivalent substance there is the matrix of the blood plasma, which is much more liquid, has a lower viscosity, but that is functionally equivalent in terms of the extracellular matrix. Red blood cells are obviously the most common. Fun fact, mature red blood cells do not have their nuclei. During their adolescent phase as reticulocytes, they dissolve their nuclei to make more room for hemoglobin and other similar substances. to increase the density of oxygen carrying the cell's oxygen carrying capacity. But also within blood, we have white blood cells and platelets. And so this is where we kind of have the notion of the immunological function of connective tissue. It's oftentimes mentioned in textbooks, but not necessarily elaborated on. The immunological function is primarily these white blood cells, which are the sisters to red blood cells. So these are the guys that are going around floating in the bloodstream, looking out for bacteria and other foreign particulates, as well as making sure that no cancerous cells develop inside of your body as well. Then the fibers, quote unquote fibers, are the soluble proteins and like platelets and such that float through the matrix, precipitate during blood clotting, and then function obviously transporting oxygen and nutrients and also removing them from your body's tissues. Here's an example. So again, this space, this plasma is the equivalent to the ground substance we see in connective tissue. We see two examples of immune cells right here. We don't really need to get into the details for the purposes of our exam, but this would be a lymphocyte and a neutrophil. The reason you can tell this is a neutrophil, it has this segmentation, these lobing processes on. So this is a fairly mature neutrophil right here, but don't worry too much about that. Red blood cells contain oxygen. I think we're all pretty straightforward with that. Another example, blood. Here we have a neutrophil. Again, we can see that lobing happening with the nucleus. Muscle tissue. We're gonna end things off with muscle and nervous tissue. This will be relatively fast compared to the earlier sections. So we have three main types of muscle. We have skeletal, cardiac, and smooth. The only one that you have voluntary control of, meaning that your consciousness can actively contract it, is skeletal muscle. And this is found in all the things that you would typically think of as skeletal muscle. Cool. What does it look like? Well, it looks something like this. So we have a nice uniform striation occurring, striations in this direction, cross-sectional functional groups that accomplish contraction. But then we have the general orientation of muscle fibers being uniform in their direction, right? They're running this way. And all of these are nicely segmented. There's no branching occurring. They're not connecting, like this one isn't connecting into this muscle cell over here. That's important as a distinction for cardiac fibers. Another note is they are multinucleated cells. So all of these nuclei would be a part of the single muscle fiber because these cells are extremely large. Most of the time, these cells are running the full length of whatever muscle they are associated with, even in very large muscles such as like your quadriceps and that sort of stuff. This is a very, very large cell. It's relatively narrow in terms of diameter, but it's very long in terms of its length. If you only had a single nucleus, then you can imagine that these distal ends, the regions over here, if this is one cell, the regions over here, the regions over here, are not going to have access to a lot of nutrients and new cellular machinery if there was only one nucleus floating out somewhere in the middle. But if we are multinucleated, then we can accomplish the metabolic needs and remove that bottleneck in its homeostasis. So all it's obviously voluntary movement, that sort of stuff. Multinucleated, see those striations, uniform orientation. That's how you will distinguish skeletal muscle. There's another slide of it. Again, you can see the striations and everything's oriented in the same direction. And then multiple nuclei that are pushed to the edge of the cell. Cardiac muscle. Okay, so here we're seeing some branching happening. So multiple cells might be connected to others via these branches. The single most distinguishing characteristic of cardiac versus skeletal muscle are going to be these things called intercalated discs, these purple little structures that are joining adjacent cells. Cell over here, cell over here. They accomplish multiple things, nutrient transport, as well as, very important for the uniform contraction of the vesicles, they allow ion transport. So this is how we can get a uniform depolarization of our ventricles to contract all simultaneously and generate a strong pumping force inside of the heart. Again, this is not consciously controlled. This is not a voluntary muscle fiber. And the only place we see this is in the wall of the heart. So the dead giveaway, if you see this on a slide, is going to be those intercalated discs. Another example picture. Then lastly, of the muscle types, we have smooth muscle. Okay, so this looks very different than the other two muscle fibers that we were looking at. We have a bunch of nuclei stacked on top of each other. We have all these long interconnected fibers, muscle fibers. Where do we see this? Well, we see this along all of your intestinal tracts as well as surrounding, it should mention somewhere, the surrounding lining of your vasculature. Think of your arteries and that sort of stuff. If we want to limit blood flow, then the smooth muscle surrounding the lumen of an artery can contract to both increase pressure as well as decrease throughput through that vasculature and divert blood flow to other areas where we will then dilate. We will relax those smooth muscle fibers and allow for a larger volume of blood to flow to a region. And then we also see this throughout the lining of the hollow organs. The walls of your intestines are lined with fairly significant layers of smooth muscle, and these uniformly contract in sequence. So imagine we have a ball of food and we want to move it forward this way. So we would squeeze here and then squeeze there, and then this ratcheting motion would occur, and it would push that ball of food through your stomach through a process called, and that process is called peristalsis. So that is what smooth muscle looks like. Trying to end here relatively quickly. Jejunum is how that is pronounced. I know it's a scary looking word, but again, relatively uniform in shape, but there's you don't have those functional cross sections that we see, those striations that we see with skeletal muscle. That's going to be the big giveaway that you're looking at smooth as opposed to skeletal. Again, no striations in this muscle fiber. Lastly, we have nervous tissue. This will be fairly quick, not a whole lot to discuss here. We'll cover this more in depth when we cover the central nervous system in class. We can broadly categorize nervous cells into two categories. We have neurons, which is what you think of when you think of a neural cell. These are the ones that are actually carrying out action potentials, the process of communication. And then we have neural glial cells, which is everything else relating to the nervous system that is not a neuron itself. Think your Schwann cells that are insulating cell fibers, immune cells that are floating out like in your central nervous system, in your brain, and that sort of stuff. Those would fall under the category of neuroglia. So supporting, insulating, and protecting the neurons. What does a typical neuron look like? Well, let's focus in on this illustration right here first, which will make this make a little bit more sense. We have the main cell body, which is surrounded by a network of branching dendritic processes. This is information inbound to the cell. This is how it is aware of what its neighbors are doing and saying. And then if it receives a signal that elicits a response that says, hey, I need to carry this signal forward, then down its axon, it will carry the signal to wherever its terminal end is, whether that's in the spinal column or to, if it's a motor neuron, to functional units of sarcoplasm, functional units of muscles, and secrete acetylcholine as the neurotransmitter to carry that signal forward, that sort of stuff. So again, dendrites are inbound signals. Axon is going to be the outbound signal from a neural cell. So here we can see these are the cell bodies, okay? And then if we're just visually looking at it, it can be relatively hard to distinguish an axon from a dendrite. I think the only really easily distinguished one is this guy right here being an axon. Of course, we see this throughout the central nervous system as well as peripheral nervous system, right? Neurons. And that is it. Congratulations. We're done with main histology slide. I am, however, going to subject you to one more example because, all right, is this working? Can't see what you guys see right now. Yes, it is. All right. So let's quickly run through some example slides as they would be presented on an exam. It's a dead giveaway, but we're looking at simple columnar. How do we know that? Well, we see all of our nuclei are relatively uniformly distributed across midpoint of this cell, and we can see each one of these cells is attaching to its basement membrane. We also have a little bit of a brush border going on along this top. And then this would be the basement membrane that they are attaching to down here. So simple columnar epithelium, pretty straightforward. Adipose tissue, okay? Make sure you can distinguish adipose from alveoli. Simple squamous epithelium of the alveoli of the lungs. Adipose tissue, these are fat cells, and these would be lipid droplets forming this space. It is relatively translucent. Even the cells are relatively translucent. And it's compared to the inner lining of the lungs, these are relatively small sacs compared to alveoli of the lung. This would be an alveoli right here. So much higher density of much more clearly visible cells with very distinguishable nuclei, and even the cytoplasm of those cells is dyeing a pretty significant pink that we can easily distinguish. And this is a very large open space cell. This is where air would be to accomplish gas exchange. Okay. I don't really like this tissue, areolar connective tissue. You can see, okay, very faintly. Let me maybe do a different color. Just one thing I will point out on this. These guys right here, oh my gosh, the color's not even helpful. But these would be elastic fibers right here. Because again, areolar has a mix of different fiber types. Blood. All right. Quick picture of blood that we took in the lab. Red blood cells, and then polymorphonuclear is the term for that lobing process on the nucleus of a neutrophil right here. To be able to distinguish those two, this is blood. That's a white blood cell in it. Here's bone. Again, the osteon would be this process right here. And then these are the lacunae, or the osteocytes, the living cells would be in. Those little tiny lines, those little striation-looking things, are tiny little tunnels, which we'll cover maybe more on another day. But that's how they communicate with each other and send nutrients back and forth. It's also how they detect pressure differentials in the fluid running in those chambers to determine whether or not the bone needs to reinforce itself due to excess strain. Cardiac tissue. Okay, so there's a relatively uniform arrangement here, but notice we have some branching going on. Okay, we have this branch, we have this branch. They're typically mononuclear. Here we have an intercalated disc. That should be a big giveaway as to what we're looking at. Okay, so that's what cardiac muscle is going to look like. Dense regular connective tissue. Okay, high density of collagen fibers running through this stuff. And it's regular because of this direction that I'm drawing onto it. If it was whooshing around, then it would be irregular. Dense, irregular connective tissue. Relatively same complexion. Oh my gosh, too many lines, guys. I'm getting too excited with it. But if it was kind of shaped like this, but relatively the same complexion, the same color staining, then that would be dense, irregular connective tissue. If it's regular, right, then we're resisting stretching motion in the same direction, aka, as an example, a tendon. Still recording. Thank goodness. I thought I stopped recording. Elastic tissue from the aorta. Okay. Same direction. Dyeing purple. Purple is going to be our elastic fibers. Cool. We already talked about fibrocartilage recently. Pseudo-stratified columnar epithelium. Okay. Our basement membrane is down right here. Beginning of connective tissue. Pseudo-stratified because it kind of looks like we have stacked. We have a nucleus here and we have a nucleus right here. So they're kind of stacked on each other, right? Well, but they're all, as we know, all of these cells at some point are going to attach down to this basement membrane. And it's fairly a dead giveaway that like this space, if you're looking at it on a slide, and then you see a bunch of connective tissue here and then a bunch of empty space up here, this is probably pseudo-stratified, right? It's not thick enough to really look like a stratified, like squamous or layering process. And then lastly, a good giveaway is we see this brush border along here, right? So this is from the trachea. Yeah, so we have the cilia lining here, okay? That brush border is a good giveaway. Simple columnar epithelium, okay, good comparison between these two, right? Stacking of the nuclei, but everything's attaching to that basement membrane with the brush border. Whereas here, we can pretty much draw a straight line along one of these cells. See that it's attaching to the basement membrane. Cool. Here, the brush border would be the microvilli, again, to increase surface area for absorption of nutrients. Simple cuboidal epithelium. This is not the best of slides, but it's good practice, therefore, because you need to be able to see the shapes and the bad stuff. I'm kind of outlining what looks like cells to me right here. This would be simple cuboidal. This is where blood would run through inside of it. Looking inside of the kidney glomeruli cross-section. Skeletal muscle, uniform orientation, multinucleated. We have to see lots of nuclei pushed to the edge of these cells. And then we see those cross-sectional striations. We already went over this. Smooth muscle. We don't see any striations in it, really. It's kind of more disorganized, certainly, compared to skeletal muscle. That's going to be a distinguishing facet. Also be typically mononucleated as well, instead of multinucleated, as we see here. Smooth muscle again. Okay, less organized than skeletal muscle, right? And then stratified squamous inside the vagina. If these cells were all dead, if there were no nuclei up here, then there's a good chance you're looking at keratinized stratified squamous epithelium, which would be your skin. But since we see that these cells are still, for the most part, living because we see nicely consolidated nuclei, this is going to be non-keratinized. And in this case, we know we're looking at the vagina. Again, stratified squamous. They get nice and flat up here at the surface. Congratulations, we are now done.